And I also would like to thank the organizers to give me the opportunity to talk of our work. And I have to just first to acknowledge that. So most of the work is actually done by my collaborators, who is Jianmin Cui in the University of Massachusetts. I'm going to acknowledge him in the end, but I just like to know at the very beginning. Okay, so BK channel is a big conductance potassium channel. It is activated by, so the conductance, single channel conductance is real large, and it is activated by both intracellular calcium and voltage. And because these two, calcium and voltage, are very important cellular signals, and the channel will just in turn regulate calcium homeostasis and the other channel function, so that it is very important in neural excitation and muscle contraction, hormone secretion, and the cardiac protection in the mitochondria. So this channel, the structure has been solved, and the channel is formed by the very similar to the voltage-gated channel, except it's not domain swapped, so that it has a pore, and formed by 4 subunits, has a pore and voltage sensor domain, and also it has a very large intracellular domain that will bind the calcium and regulate the channel function by calcium binding. Okay, so now the very important and the prominent characterization of this channel is the allosteric gating of this channel. That's in a very different mechanism with some canonical voltage-dependent channels, such as the Shaker potassium channel. Here shows Shaker potassium channel, and if you look at the Shaker potassium channel, the open probability at very, very low voltages, and it goes to 10 to minus 7 open probability. And that, you can see that the voltage dependence is, in this semi-log plot, is almost linear all the way. So that it means that the channel is very tightly controlled by voltage. And we call this obligatory control by voltage, so that the voltage sensor movement will just open the channel instead of closing the channel. It opens the channel, and vice versa. If the channel opens, the voltage sensor has to open, so that there is this very, very tight control. But if you look at the BK channel, you measure the open probability to very low open probability, 10 to minus 7, and you will see that it deviates from this linear relationship. Instead, the channel seems to be always open, even when you go to very negative voltages. That means that at very negative voltages, the channel is no longer controlled by voltage. So the channel has an intrinsic opening. It just opens, closes without caring about where the voltage sensor is. So this intrinsic opening is very important, it turns out. Although the intrinsic opening is really small, 10 to minus 7 in the open probability. So because of this intrinsic opening, you can only model this channel or understand the opening of this channel by that the channel has this open and close that is not voltage dependent. And on the other hand, the voltage sensor movement would just each voltage sensor movement, there are 4 of them because 4 subunits, would just increase the open probability by a factor D. So that what this means is that the voltage sensor movement only promotes the channel opening, make the channel more preferable to the open state by this allosteric mechanism, not the obligatory, but it's allosteric. Okay, and this, likewise, the calcium-dependent, and this is voltage, and this is a calcium-dependent activation of the channel is also like that. So there are 4 subunits. Each subunit has calcium binding sites, and then each binding sites occupied would increase the open probability by just allosterically promote the channel opening. So if you combine these two models, and you'll see that the channel opening is consistent of those major three steps. So one is the intrinsic opening, it opens and closes, and voltage just regulates this intrinsic opening, and the calcium likewise regulates intrinsic opening independently. What it means is that if we can just hold the voltage sensor at very negative voltages at resting, the channel can be activated by calcium, and vice versa, channel can be activated by voltage in the absence of calcium. So this is a very flexible system if you think about it. If you think about the shaker channel, which is obligatory, you can never activate shaker channel independently by calcium or by another ligand without the changing voltage dependence, because the voltage sensor will just move when the channel opens. So they are tightly coupled. In this case, they're not, so that the channel can be activated by both voltage and calcium. So this intrinsic opening is really a defining mechanism for the channel to open by both voltage and calcium. Okay, this is very, very important. And another thing that is important, this intrinsic opening, is illustrated by this BK channel opposite. That means that it's a BK channel mutation that is associated with a very severe neurological disease or a disorder. This patient presents the dyskinesia, or the movement disorder, and autism spectrum disorder, and also learning disability. Although there's no epilepsy present in this patient. And if you look at this, the mutation, what it does to the function, you see that the channel seems to be activated by less voltage, and at both calcium. And if you look further, you find that the channel would only increase the intrinsic opening. This mutation only increases the intrinsic opening, and it doesn't change the calcium dependence. It doesn't change the voltage dependence. So the voltage sensor movement is not so much changed by this mutation. So what it illustrates is that if you just change the intrinsic opening a little bit, that it will just cause very severe neural disorder in patients. Okay, so now I just want to make sure that I stated that this intrinsic opening is very, very important for this channel. Then how does this intrinsic opening come about? What is the mechanism? So what I'm going to show you in this, from now on, it's not my work. It's mostly my collaborators' work. Although we talk a lot, we collaborate. So, but I just want to make sure that this property, the intrinsic opening, we think is actually derived from a very specific structural property of the channel. If you look at the channel with the calcium-free and with the calcium bond, supposedly this is an open channel, this is the closed channel, you don't see the pore closing. So you see that the pore inside of the pore wide open, not like other channels or voltage-gated channel. We know that it is just the intracellular side of the channel that would intracellular side of the channel here, that there will be the bundle crossing that physically close the channel. We don't see that. So no one actually has the structure would show that or see that. So. But if you look carefully about this change in the metal bond and the metal free, supposedly open and closed, you will see some subtle changes. And for example, there are those hydrophobic residues that will, in the open channel, in the metal bond channel, they will just form a patch of the hydrophobic patch that is not facing the pore, inner pore. But when it is metal free, these hydrophobic residues will just point to the pore, so that the increase the hydrophobicity in the inner pore. And if you look at the size of the pore, inner pore, you see that in the metal-free channel, that the pore becomes a little narrower and a little longer, and also now it is a little more hydrophobic. And these changes turns out to be very critical for water molecules in these narrow pores. So these changes actually will drive the water out, so that it's called de-wetting of the channel pore. So here is the simulation that shows that these are using one kind of a model of, or force field and the water model, and this is using another set of force field and water model. But basically, you use different method to calculate, and you see that the water, the pore will just dewater, or the water becomes just driven out of the pore when the channel is in the closed state, and those were the simulations that it shows. Okay, so now if you look at this after the de-wetting, you will see that the free energy profile for potassium going through the pore, permeation through the pore, the energy will become much larger than in the water filled pore. So that it's understandable that when you don't have water, ion has no place to be solved, so that it doesn't go through. So the pore actually the energy becomes too high for the potassium ion to go through. That's how the channel is closed. But also after this, dewatering or the de-wetting of the pore, you find that the pore also changed further, that it becomes even narrower. Although it is sufficiently wide, if you had water in it, the ion should just not be stopped. So it is just the dewatering or de-wetting of this pore that makes the channel to close. And this is something called hydrophobic gating. Okay, so it's not conscious to the physical gating. So this was actually the work published previously. Okay, so now how does this have anything to do with the intrinsic opening of the channel? So we find that if you have a hydrophobic gate instead of a physically closed gate, the energy that free energy prevent ion to go through is finite. Okay, so this is about the kcal per mole, the energy here. And if you just think about it, with this energy, if you give a sufficient driving force for the ion, and sometimes that the pore will just occasionally have water in it, will drive water into the pore, and the pore becomes leaky. So this is just the calculation based on this 8 kcal per mole, and you will see that the open probability would be 10 to minus 7th, which is very, very close to where we measure the intrinsic opening, 5 to 10 to 6, 10 times 10 to minus 7. So what we did was then made a simulation of this profile, of energy profile for potassium ion to go through the pore at different voltages, and then we get the current just plotted. This is the experimental data that we measured, the open probability, and this is the open probability we just actually calculated from MD simulation. They quite nicely overlap with each other. And this was also done with another method that is just to use the profile, energy profile for a potassium ion to go through at 0 millivolt, and then we add voltage to the system, and then calculate how would the current will be dependent on voltage, and that actually gives rise to some voltage dependence, as we saw in the experiments. So those two methods both come up with something that is quite similar to what we observed in the experiments. And we also made the simulation of another channel, that is, this is a channel we call core MT channel. This is a BK channel that the cytosolic domain was completely deleted. And so now, so this channel now is no longer calcium dependent, but it's a voltage dependent channel. But it is, the permeation is very much like a BK channel. And then because there is no large cytosolic domain, and this channel becomes more flexible. The changes with time, so that we calculate this again, and we also measure the limiting, the intrinsic opening. The intrinsic opening actually increases because of the flexibility, and if you make the simulation to use this hydrophobic gating simulation, you find that the simulated data also matches with the experimental data. So these experiments promote us to think that the intrinsic opening of BK channels, it is very important for allosteric gating mechanism, and this is basically derived from this hydrophobic gating of the channel. The inner pore of BK channels remain physically open but undergoes hydrophobic dewetting. So there is that it overcomes this. Overcomes this. The leakage overcomes this vapor barrier. Okay, so I would like to point out that to just connect the intrinsic opening with this hydrophobic gating actually gives rise to a way to study hydrophobic gating, because when hydrophobic gating happens, the channel supposedly closes. How do you measure a closed channel? And that, initially we thought that we couldn't measure that, but now when you have this leak current from this closed channel, that gives a way for us to measure the property of the hydrophobic gating when it is closed. So I'll, in the last, that's very important, and I'll just acknowledge that the work, the simulation work was done in Jianhan Chen's lab in the University of Massachusetts, and by this very talented postdoc, Zhiguang Jia. And the experimental work was in my lab was mostly done by Guohui Zhang, who is also a postdoctoral fellow. The mutation that is associated with the patients were actually communicated to us by a hospital in London, and to Dr. Mikati, who is a neurologist in Duke University, and through also the collaboration with Huang Heyang, also at Duke University. Thank you very much for attention.
Thank you very much for introducing such an interesting idea. Thank you very much. Okay, so the paper is sure. Open for discussion, comments?
I'm sure you must have said it and I missed it, but where is this mutation? The mutation that you showed in the beginning that remained leaky at very low. Where is it?
The mutation is in the very end of the tail in that cytosolic domain. And we think that this mutation will somehow changes the structure of the cytosolic domain to pull the channel open a little more.
Mm-hmm. So, in this respect, why do you exclude that it's not just that the linker is kind of less flexible? And even in the absence, probably you remember certainly this paper by Carl Maggleby's lab, that the linker, even in the absence of calcium, has some sort of elastic potential energy on S6.
You mean which leak? What kind of leak is? So the intrinsic opening is.
Yeah, so that this mutation basically permanently changes the elastic properties of the linker, and by this way, it is somehow related to the calcium pathway, but it's just a passive stiffening of that linker, as has been.
I see. Yeah. So Carl Maggleby's lab actually showed that the linker length is dependent. Determines the voltage and the calcium sensitivity. We think that maybe that the linker length could change the intrinsic open probability, but we don't know about that. no one actually studied those different linker lengths on the intrinsic open probability. But presumably, it could also increase or decrease the intrinsic open probability because of the pooling of that pool. Yes.
Very interesting talk. You introduced two concepts, leakiness and channel open probability. And in my mind, they're different things, but the way you described it, they sounded like you were talking about the same thing. So I feel like a leakiness is when your channel is closed and some ions get through, whereas open, it's moving a conformational change and more ions go through. And you should be able to tell the difference between those at a single channel level anyway. So I'm wondering, when you first showed the graphs of open probability, can you tell if that's a conformational opening or if that's leakiness?
That's a very good question. The leaking current that we saw was also the single channel. So I particularly left it out. But so we measured the single channel opening in a macro patch with many channels, and then we measured the opening to get the overall open probability. So that was measured by single channel opening. And you see the single channel opening in there is very similar to the single channel opening in the voltage dependence and the calcium dependent channel opening, except that it is very, very brief. And it's it's not like with calcium and voltage dependent activation, you have much longer opening. You can say that it is a leak. If you just look at a single channel, you can say this is a leak, or this is just because it's at a very negative voltage, so that it becomes very brief. So all we can see is that at a negative voltage, the open probability is no longer dependent on voltage. So that's why we call this. Initially we call this intrinsic opening, but we think this is a leak.
I was thinking you could differentiate by magnitude, but I think I need to ask you about this
magnitude
afterwards.
of single channel
current, yes.
So the leak, so-called leak, is that one pore very transiently filled with water because of the driving, and then the ion will just go through that water, so it's just acting like the channel is open. We cannot tell the difference between, yeah.
Wonderful talk. Thank you so much. So I'm a little bit confused about the construct without the CTD. So you said that the open probability of the leak current is higher in that construct. And you said that the reason for that is because the S6 is more flexible. So to me, in my mind, the S6 being more flexible would allow this channel to close more. And I would expect this open probability to be lower in the limiting slope.
Yeah.
What do you think about? And then I actually was going to follow up on what Ben was saying, that maybe this particular mutant may allow you to distinguish between the two mechanisms that he was talking about, because you have a higher open probability, you can look at the single channel conductance and see whether there's a difference.
Well, first of all, the single channel properties were not so much difference, except that it's just opened a little bit more with higher probability of opening. And secondly, it depends on how you think about it, that whether it should be more flexible or to open more or open less. So if it's more flexible, so that it just breathes a lot more, that so that it has more chance to be leaked. That's what we think. And so obviously we don't have any structure or any data to show that. So this is what maybe I shouldn't say it's more flexible. I just want to say that the simulation matches the experimental data for whatever reason. So that the reason that it's more flexible is made up.
Right, but just to follow up, I mean, you were saying that during the MD simulations, the S6 actually narrowed down a little bit. So I would expect that the reason it didn't narrow the way which you would expect it to happen if you remove all the water from the pore, and now you have this hydrophobic seal going on, that it's actually kept open by something else.
Yeah, so actually, what the simulation shows that it just the energy profile for potassium to go through is actually reduced a little bit, and that is why you get the higher open probability. That's from the simulation. Okay, we can discuss a little more.
Okay, so the last question.
A very short question. In the A532B mutant, you showed there is more leak opening. And is there any point mutant which lacks leak opening completely?
To make the leak a little, or to make the intrinsic opening a little lower.
To remove the leak opening completely, like shaker.
We don't have any mutation that completely removes it, but we do have mutations that reduces the intrinsic opening by a lot.
And the position of the mutant matches with your hypothesis.
If you simulate that,
Okay.
that is, yeah.
Okay, the final question.
Yes, hi. A very interesting observation, but I'm wondering what would be the physiological consequence of these openings at minus 100, close to EK, especially since cells will not often be at that voltage.
Absolutely. No, you don't see that low voltage. And so you don't see those, the leak itself. But as I said in the beginning, that the leak of the intrinsic opening is very important to determine the voltage dependence and the calcium dependence. So that you see all the time in physiology, and as the mutation shows that if you change it a little bit, then you get the disease.
Okay, thank you.
Yeah.
Okay, thank you very much for a vigorous discussion. Thank you very much.
Thank you.
I would like to close the second session. Thank you very much.